giving us opportunity to share with you our GENA-LM family of DNA language models. I will start with brief introduction into the field. As you know, to effectively apply machine learning approaches, it's better to have large data sets. A single human genome with 3.1 billions of base pairs printed in font size 4 bounds into 130 books with 78,000 pages. And sequence read archive reports that normalized format data is projected to grow to 33 petabytes by 2023. So in genomics, we are in the case with large amounts of at least unlabeled data, and the machine learning methods can help here to analyze them. There are lots of complex dependencies in the DNA data. It's well known that functional genome elements like promoters and enhancers, splice sites are directly controlled by DNA. Here we can see how gene expression can be affected by natural variations like positioning of transcription start site, protein interactions between transcription factors, or protein DNA interactions. We encounter long-range dependencies, loops, spatial arrangement importance, and it makes analysis of such kind of data very complex. However, there are successful applications of machine learning to large-scale genomics data analysis. For example, DeepC effectively predicts non-coding variants, splice-I for splice site predictions, and former for gene expression. However, these are specific methods for each biological task, and as these models are specific, it's not clear how to use or adapt them to another tasks without retraining on new task-specific data. Transfer learning became go-to technique in natural language processing and computer vision. It allows to reuse large compute and general knowledge gained during pre-training on large collections of unlabeled data to faster adapt to downstream tasks. With fine-tuning, few-shot or zero-shot approaches, in-context learning, prompt tuning, and many other techniques, getting finally task-specific models as a result. We can go from natural language to the language of nature. Here's a well-known NLP BERT model and DNA BERT published in 2021. These models look quite similar. The difference is only in what kinds of data is used as input. Here we use natural language texts, and here sequences from DNA. What are tokens, sentences, and documents in the DNA? We can, in this question, we can closely follow NLP data pipeline. As text data, we take all 3.1 billion base pairs from human genome and randomly split them on sentences with 500 to 1,000 base pairs chosen randomly. Then we collect these sentences into documents, and multiple documents build our training corpora. This pipeline was proposed in the Big Bird paper, and we use it in our GENA models. What are the tokens in DNA? There are several most common approaches. The first one is to use single token nucleotides or characters. More common approach to bioinformatics are k-mers, and from NLP we can use tokens that are trained on available data. Each token is comprised of multiple nucleotides. The first two approaches lead to models with sequence length equal to number of nucleotides. Here tokens consist of multiple nucleotides, and as a result, model sequence length in base pairs can be longer than just number of tokens. In case of transformer models, sequence length is an important limitation, as compute and memory complexity of self-attention layers has quadratic scaling. In GENA-LM, we use byte pair tokenization. It gives us ability to cover about 4,500 nucleotides with 500 tokens and 36,000 nucleotides with 4,000 tokens. Tokens in the vocabulary may vary in length. Here's the distributions of tokens length. Median length is about 9 base pairs, and we analyzed long tail of this distribution and found that some of them represent well-known repetitive elements such as lines or simple repeats. Such tokens in vocabulary helps us to represent such repetitive parts of genome with a single token, squashing dozens of nucleotides into a single token. During pre-training, we add special tokens CLS and separator, and some of the tokens are masked, and model is trained to predict what is covered by these masks. Basically, it's a masked language modeling objective. What data do we use for pre-training? We use T2T genome assembly, which was published in 2022 and has 10% better genome coverage than previous builds. Other transformer-based DNA models are trained on less up-to-date builds. For example, Big Bird uses version 19, DNA Bird and nucleotide transformer use version 38. We also augment data with reverse complement sequences, given 2 times more data. We add augmentation sampled from SNP's mutations from 1000 genomes project that gave us 10 times more data. Some of the models we trained not on human-only data. We add genomes of about 30 selected species from GENOMAD. In total, for multi-species models, ratio between human and non-human data is about 1:1. As a result, we collected 1.1 trillion base pairs to train our models on. Currently trained DNA language models are based on transformer architecture, but differ in tokenization, in sequence length, and in pre-training data, also in model sizes. Nucleotide transformer has models with larger number of parameters. However, GENA-LM models offer sequence length, much larger sequence lengths. In GENA-LM models, we made some changes into transformer itself. We incorporate pre-layer normalization. It changes order when to apply layer norm layers after multi-head self-attention or before multi-head self-attention. It was shown in NLP papers that it makes training more stable with different batch sizes and learning rate schedules. For longer sequences, we use sparse attention with diagonal, global, and random attention patterns. This combination of patterns was proposed in the Big Bird paper. Sparse attention and its effective implementations, for example, in DeepSpeed or Triton, save lots of compute in self-attention layer, in self-attention layers. We also add rotary positional embeddings. It was shown that they help to achieve better quality than learned absolute embeddings for positions. Rotary embeddings can be used with effective sparse attention implementations because they support relative distances that does not require to learn full square matrix of positional embeddings between all possible pairs of distances. As a result, rotary embeddings got wide adoption in NLP models, and we also tried to use them. Here's a list of all models in GENA-LM family. We made all our pre-trained models with different lengths and with different sizes, 12-layer models, 24 layers models, with sparse attention or without sparse attention. And also our models differ in pre-training data. Some models are trained on human-only data, some models are trained on additionally on multi-species data. And all these models available with Hugging Face transformers. You can find them in our organization, IRE Institute. And next I'm giving a mic to Veniamin to cover downstream tasks and GENA applications. Okay, I'm stopping sharing screen.
Thanks. Okay, the same slide, just with a different background. And with it, I would like to start the second part. My name is Benjamin Fishman. I'm going to follow up with a brief description about how you can use these GENA models for different downstream tasks in human and probably other species genomics. So I will start from a challenge of predicting promoter activities of DNA sequencing. In machine learning terms, it would be a classification task where you would like to classify sequences into promoters, technically those which are just sequences upstream of transcription start sites, and non-promoters. And to make this a bit more interactive, I will be showing some code snippets, but since it might not be very easy to follow just from slides, I also put a QR code here, which will take you to a Jupyter notebook with the same code. So after talk, you can, if you want, study it thoroughly. So first step to solve this task is actually to construct a data set. And for this data set, you only need a simple CSV file with the sequences and labels, which is promoter present or promoter absent, 0 or 1. And then using the standard Python transformers model classes such as dataset, you can just in a few lines construct a dataset from this file. I will be actually showing lots of examples of how to use GENA infrastructure with these transformer classes, and they are well documented. So please follow to the documentation of these transformers to know more about these steps. So after constructing this data set, you would need some pre-processing steps. For example, you would need to rename the labels because if they originally were called promoter presence, for working with standard classes from transformer libraries, you would need to relabel them as label. And also you would need to process the sequences, to pre-process the sequences, because as Yuri already told, we tokenize input sequences. So you would need to use our tokenizer, which is also available on the Hugging Face. I will show you links in the end. And here is how you can do this using the standard classes from transformers libraries. So by this operation, you process the sequences to obtain tokens. And then you need one of our GENA models to get some meaningful embeddings of these tokenized sequences. But before I show how to use our GENA models, I would like to explain in general what you can get out of a language model, DNA language model like this one. So typically when you apply tokenization to your sequence, you convert into a set of tokens, and you also include some service tokens like CLS and SEP tokens shown here. And after this, we'll go through the neural network, you will get an embedding or vector relevant for each token. And there is no unique or good answer for all tasks, which one of these many vectors you should use for downstream application. So all of them contain some meaningful information. There are also some standard solutions. For example, for whole sequence classification, the standard solution would be to use a vector which corresponds to the COS token. And we will follow now the standard solution, but of course, it's up to you to modify it for your specific tasks. So, as I said, we would like to use this output, the COS output from the model, and then put a simple, fully connected layer on the top of it to get a label, single label, the absence or presence of promoter from the sequence embedding contained in this output. So how you do it technically, it's again just few lines of code. You use a standard class Big Bird for sequence classification, which is from Transformers library, and you indicate that you are solving classification task, and basically that's it. But note that here you would need to initialize this object with our model, which is deposed in on Hugging Face, and you will need to specify the number of labels. It's 2 in this case because we have one label for presence of promoter and another label for the absence of promoter. So after you have this, you can start a training loop, which is also can be done using standard tools from the Transformers library. And you can see here that the training is ongoing, and I would like to highlight that the benefit of pre-trained models that you don't need too much resources for downstream tasks compared to solving this task without pre-trained model. So after you get the trained model, you probably would like to make an inference for your biological problem. And here again, I would refer you to standard methods, and we put some examples into this Jupyter notebook to show how this inference can be done. So back actually to our work, let's see how good these GENA models are in the promoter classification task. Here I am showing different tasks, and the difference is the promoter sequence length. So we can extract just 300 base pairs upstream of the transcription start site, or 2 KB or 16 KB, and all of these sequences were fed into the one of GENA models or into a baseline DNA BERT model. And the first that probably you see already from the results is the longer you have the sequence, the better is performance. But also within the group of sequences of the same length, dependent on the model, you can have different performances. For example, for very short sequences, DNA BERT shows the best performance here. And also between GENA models, you have the best model, which is BERT large. And this is a model which has shorter inputs, but more parameters. But when we go to longer sequences, first, all the models outperform the DNA BERT. And second, for medium-sized sequences, we still have this BERT large model, which is the best. But for ultra-long sequences, which are 16,000 base pairs, we have another models which have less parameters, but due to sparse attention, they have longer potential input. These models, they outperform the BERT large model. So depending, for example, on sequence length, and as I will show later, depending on other parameters of your task, different models could show the best performance. So probably try different models in your specific application to find the best one. Well, this was the classification task. We can also similarly solve the regression tasks. For example, polyadenylation signal strength prediction task here is actually a task to say how good is a sequence in a determination of transcription stop and polyadenylation, meaning how often the RNA polymerase will stop on this particular sequence and stop transcription. And this is a quantitative trait, and you can very similarly use the same code to solve this. You only need to change the number of labels, because now we have only one quantification, meaning that the number of labels is 1. And this is actually, again, I'm pointing you to the documentation of this transformers code, because it's well documented here in the manuals how you can switch from classification to regression task. And back to our results again. So here we have all our models outperforming the DNA BERT. And also now on this slide, you can see this horizontal line, which corresponds to a published baseline on this specific task, which is in this case a convolutional neural network. So you see that all transformer models outperform it, and GENA shows superior results. We also have can have some multi-class challenges, for example, prediction of splice sites. And here, sorry, anyone has a question now? Okay, then I'm going on. Here, for example, the challenge is for each token within the sequence to predict whether it's a splice donor, like this point, splice acceptor, like this point, or none of them. What it means from biological point of view, it's a very important task, because you probably know that in human genome, the genes are not continuous, and they are separated by fragments called introns, which are removed by molecular machinery during processing of RNA molecule. And it is to understand the final structure of the RNA and which protein it will code, we need to know these junctions, which are called, as I said, splice donor and splice acceptor sites. So how can we do this with GENA? Again, we use standard libraries, and we just change the number of labels. So it's again very, very similar code to perform this. But the problem is that we also need to switch from the whole sequence classification or regression to a token level classification or regression, classification in this case. And for this, again, we can use standard classes. So we only modify the class, and we solve the token level challenge with basically same code here. So what we have as a result, now it's not as optimistic. GENA performs quite well, but not on the level of the state-of-the-art convolutional neural network published by Illumina in 2019, Splice AI. So the performance is slightly lower even for the best GENA model. And also, I would like here to highlight some limitation of GENA model, which is due to tokenization. Basically, for splice sites, you actually can go in biological data, you can go beyond the token level to individual nucleotides and specify exact nucleotide position where the splice site occurs. But since GENA operates with tokens, we cannot do this. We only can say whether the token contains the splice site or not. And although most of tokens are quite small, it is still a different level of resolution compared to the nucleotides. We are working now, and I will show you our future plans in the end, but I would say now that we are working on solving these problems, but if you will use our solutions, which are currently published, then probably you will have this limitation of the resolution. Okay, so now I will briefly show several more tasks that we can solve with GENA. For example, this is a very well-known task in chromatin biology. It's a prediction of chromatin binding sites by different proteins, occurrence of different histone marks in various sites of genome, or DNA's accessibility, which means how the DNA is accessible for proteins to bind it, to read it, and to somehow manipulate with this. This is a data set which is called deep sea data set, and there are several challenges in the field which are based on this data set. And these challenges are prediction of different epigenetic marks, as I said, separate for transcription factors, for histone marks, and DNA's accessibility profiles. And you can see here that, again, basically for majority of these challenges, the GENA shows very good results. But again, for some challenges, one model would show the superior quality. For other challenges, it will be another model. So probably to find the best model, you should try several of them. And again, we can see that increasing the lengths of the context, in this case, it will be sequences flanking from both sides the region we are interested in. So increasing this context, we will have the higher and higher power of prediction. And with the longer contexts, we are able, with the best models, to outperform both a solution, an original solution for this data set published in 2015, and also the very new nucleotide transformer solution, which is a preprint published just a couple of months ago. This is shown in two horizontal lines here. But also it's interesting that for some features, increasing context actually gives lots of gain in performance, but for others, it's not the case. The level of performance is still the same. And we're interested why actually, what is so special in the features which gain this increase of performance when we increase the DNA context around. And here we compared the score for 1 kb context and 8 kb context, and we found that there is a set of features which perfectly overlaps with the broad histone marks that gains performance for longer context. And what are broad histone marks? These are special modifications of histones which spread across long DNA regions. They are not just found in a small sequence of several hundreds of base pairs, but they are found in kilobases or hundreds of kilobases, continuous regions across genome. And what it means, it actually means that these marks are probably not encoded biologically by some small specific sequence somewhere within this broad region. So probably the biological mechanisms implied in the deposition of this signal is somehow rely on different sequence markers distributed across a long region. This is important for us to understand further what are exact biological mechanisms underlying these features. And the final example I would like to show is a prediction of Drosophila and cancer strength. And why I would like to close with this example, because all the previous data was human data. But actually, there are many challenges which are related not to the human genome, but to the genomes of other species. And here we benchmarked this Drosophila challenge, and we found that actually all genome models, doesn't matter whether they were trained in a multi-species setup like here. This one is the model which was augmented with genomes from other non-human species, but also other models which only include human data. They are equally good in solving this challenge, or even actually models trained on human data are better than model trained on a Drosophila and other species data, including human. So this is a bit surprising for us, but this shows that you can easily transfer this knowledge learned by GENA models from human genome to the genomes of other species without repeating this pre-training on the specific genome of your favorite species. So I would like to conclude with this and just summarize what we have already told, that GENA is a foundational DNA language model with the largest input size, and that GENA can be used for various downstream tasks. I showed you examples, and also trained on human data, it can generalize to solve tasks specific for other species. For many tasks, although not for all of them, the performance of GENA is either matching or exceeding that of prior models, whether they are task-specific or universal, such as DNA-BERT. And finally, the practical advice is the choice of optical model depends on the task. So just try different models which are all publicly available. And finally, our future directions is to increase input sequence length even further. So we are working on models with much larger input lengths, about 100 of kilobases or 1 million of nucleotides. We are working on understanding some biological interpretations of GENA results, as for example with histone marks that I showed you. And of course we would like to add much more different downstream tasks, and for this we definitely need your help. Please use GENA in your research. We will be happy to answer questions. I will show you links to our GitHub where you can open new issues if you have troubles with using GENA. And also, we are planning to perform some webinar with questions and answers. So please let us know in a Discord, for example, if you have some specific questions which you would like to be answered during this meeting. And with it, I'm showing you some links to our model repository, to our GitHub, and to the archive where you can find the manuscript with the data I told about and Yuri told about today. So thank you very much. And together with Yuri, we would like to answer your questions.
Hi, thank you very much for the fantastic presentation. I think other people will also have other questions in the chat maybe, but in the meantime, I'd like to ask, I come from a protein background, and there has been significant success in leveraging protein language, unimodal, sequence-only protein language models, especially when coupled with some representation of a structure. So techniques that leverage multimodal data type. I am not really up to date with regard to the state of structure availabilities for entire chromosomes, for example. Do you see the sequence-only paradigm continuing in the future? So sticking to a sequence-only approach, or do you also see structures being integrated, especially for downstream tasks in the short term or medium term?
Well, there are very interesting solutions in this field predicting the DNA structures, not directly structures, but the data which is called high-C data, which describes the pairwise contacts between DNA fragments. And there is a very nice work by Joe Padenberg and Kelly's group from US, and there are actually several of them, but to highlight one, for example, who show that it's possible to use a sequence input to predict the structure, which means that the GENA model probably can infer the structure in a similar way. But I totally agree that it would be interesting first to benchmark the GENA in this challenge to see, because it's theoretically possible, but we haven't checked whether it's in practice it happens. And second, maybe to explicitly add some of this information, but this probably depends on a particular downstream task, whether it's needed or not.
And do you think for this particular, structure prediction task, given that at least for proteins, we've seen similar models being highly successful, for example, ESM-fold, leveraging a structure model being applied on top of ESM, do you think significantly longer context sizes, like you mentioned towards the end of your presentation, being a necessary requirement? Because as far as I understand, dependencies can be and interactions can be extremely long range in genomics. And do you think then that those kind of context sizes will be required for genomic structure prediction, or do you expect even a 1K context size to be sufficient for structure prediction still?
Well, it's not 1K now. It's several dozens of Ks what you can input right now into the genome, but I think it's still not enough. So it should be more. But technically, it's challenging. So that's what we are working on. Of course, we would like to increase it to, I would say, so for most biological tasks, something like million or several millions nucleotides should be enough, because we have some experimental evidences that the regulatory elements very rarely are located more than several millions base pairs away from the target gene promoter, for example. But still, we are not reaching this distance yet in models.
Fantastic. Thank you very much. Maybe we can check if anyone has any questions or other topics they'd like to ask. I think I see the
Yeah. Hey. So I was wondering, does this have applications to protein design? Possibly, and also like the two problems that I'm interested in, one is generative protein design, and the other is, can this possibly be used to predict impact of mutations on function, protein function?
Well, it will be just an idea which is coming into my mind, because we don't have anything done particularly in this. But what can be probably done is to predict binding sites for DNA binding proteins. So probably from the embeddings which this tool produces, you can somehow train another tool which will predict whether wild type or altered protein can bind a specific segment of DNA. This is actually what we do in a chromatin challenge.
Okay. What's the chromatin challenge? Is it
chromatin challenge is here a data set of known binding sites for very different chromatin proteins. For example, histone modifications, but also transcription factors, because although they have specific sequence recognition motifs, these motifs are usually very small and they do not fully determine the specificity of binding. So in many places you will find this protein bound when the motif is not absolutely equal to the consensus, and also in many places where you have a good consensus, you don't have a protein bound. And this tool can solve the challenge to predict where specifically this protein is bound. But this is done using some experimental data. So we already know binding sites, and based on this, we can learn in a portion of genome, we know binding sites, and based on this, we can learn binding in other genomic sequences. And what came into my mind when you asked your question about somehow adding the layer of knowledge about protein sequence here is, what if we have, for example, a very new protein just designed with something in mind, and we want to answer a question where this absolutely new protein will bind in DNA. So for this, probably these GENA embeddings can work.
Okay. And I joined the talk a few minutes late. I was wondering, could you briefly tell me? I'll read the paper for later, but could you briefly tell again, perhaps, what was the kind of training data used to train this language model?
Yura, do you want to add to this, or should I? I can open the slide.
Yeah, open the slide, please.
Yeah, here.
Yeah, we used human genome assembly named T2T. We augmented to have much more than 3.1 nucleotides in the data set. We add reverse complementary sequences. We add some augmentations sampled from SNP's mutations taken from 1000 human genomes project. And we also tried to add multi-species, like we tried to add genomes from non-human data. And basically, these are three main sources of our pre-training data.
So one question I had was, if the downstream tasks are all human, don't you think adding non-human data in the training would actually make it less performant on human downstream tasks? Or are there like conserved patterns across species which like I was just trying to understand why add non-human data in the training?
We tried different benchmarks for human only data and for not only for human data, and we saw that this adding of multi-species data does not always give us some benefits for human only data, as for example on this slide.
Okay, thank you.
But that was a hypothesis that it's like intuition, maybe from natural language processing, that multilingual data can improve, we can transfer from one species to another species knowledge gained during pre-training. But we didn't much of experiments on different species. How does it affect? How does this pre-training affects? We have only Drosophila data in one of the experiments. Yes, Benjamin.
Yeah, that's exactly like this. So we thought that it would help for, first of all, for non-human data sets, and might help for human data sets as well. But the results show that we were wrong. Well, that's how experiments work.
Right.
Thanks for the talk. It was really good. My question was if you could expand a bit more about the biological interpretation. I was particularly curious about how would you approach because the way I understand it is usually we'll use like feature attributions to like pull out motifs, things like that. How does that work with tokenized input?
Well, do you mean this particular results, or in general how we are going to interpret this this data?
I guess particularly interested in doing motif discovery, extracting motifs from language models like GENA-LM that take tokenized input.
Mm-hmm. Okay, so, well, I don't think it would be much of a spoiler to say that we are already working on this, and we have some preliminary results which we didn't put into the manuscript because we want to finish this completely. But basically, how we do this, we use the techniques to check what is the what are the most important tokens first, and then based on these tokens, we see, as in the classic, for example, chipsec experiments, where you have lots of locations for binding of a specific protein. Here we have lots of tokens, and we look for some similar motifs within them. But as I said, this is a limitation, and we understand this, because we would like for here, I think it's not a big problem, but for many tasks, and maybe even for this one, it would be very nice to go beyond token level to individual nucleotides. And we are working on solutions to this also. But and to add to this, I would like again to say that the tokens are actually small. So the mean token length is 9 base pairs, which is similar size as a typical transcription factor binding motif. So in this particular case, it's probably not a big problem. It's solvable, but for others, could be an issue.
Thank you.
I have a question about adding mutation into large model. How do you add mutations? Did you produce something like personal genome with personal mutations and just partition it into parts and feed the model?
No, not exactly. You mean this augmentation with
Yes.
human variants? Yeah. So for in the model, we don't have for each run, let's say, we don't have to process all the human genome. So we split it in chunks, which are called documents, as Yuri showed in the beginning. And within each chunk which we process, within each sentence that we process, we include one or several variants which are present in the data set. And in this process, we only use variants from one individual each time. So it will not be a combination of variants originating from different individuals. But in our data, we cannot resolve haplotypes. So from the biological point of view, it's still not absolutely accurate, because we have a kind of mixed haplotypes here. But at least we don't have mixed variants from different individuals.
I think we have one more question from Andrea Rosario.
Thanks. Thanks a lot for the talk. Yeah, it's a very interesting topic. I also had a bit of that question about how did you augment it, but I think it was clear. My question was more on how, so on the data that you trained, did you consider using the 1000 genome project? Like, did you consider using more genomes than just the reference genome? And my thinking was just on, so the results that nucleotide transformer obtained were much better with the 1000 genome than just using the reference genome. So yeah, just thoughts on that.
Thanks, Andrea. So let me check whether I understood you correctly. The question is whether we want to add more augmentation beyond these variants we already included.
No, sorry, no. My second question was more onto if you consider using the 1000 genome project, so that contains 3000 or something samples of genomes, and not just using the reference genome.
Yeah, well, we did this. So we did this because we have the SNPs collected from the 1000 Genomes project.
Oh, okay, so that's where it comes from. Okay, so my confusion came from when you say that it just used the reference genome. So, okay. So you use the 1000 project genome as pre-training data.
Well, that's probably confusing because we have too many models. I agree. So it depends on which model you use. For some models, we only use reference genome T2T. For other models, we also augmented with this 1000 genome data set. So you can explore different models. Their performance depends on the task.
All right. So when you say you use a 1000 genome SNP, so you didn't use the whole genomic sequence?
Well, we input in our experiments, the input length is limited. So we never input the whole genome, and we put all these augmentations from all individuals in a one data set. So we pass through the model chunks of the DNA which belong to different individuals and contain these individual specific SNPs. Do you understand that correctly your question?
No, sorry. My question was just that when you say you added the 1000 genome SNPs, so you added that information, but you fed to the model the sequence itself. No, you didn't just fed the SNPs, right?
Yes. We take the sequence, we change the nucleotides according to the data from 1000 genomes, and then we pass it through the model.
Right. Okay. Yeah. So I think it's just a matter of wording. My other question was, if you, on the downstream tasks, did you consider looking at how the model might perform with prediction of deleteriousness of the variants, or pathogenicity, in other words?
Well, it would be a great task, but for this we need a same great data set, because we tried some. Well, the probably, from my understanding of biology, of how it works, it should be some regulatory variants, because the variants which change the protein structure probably cannot be captured by this model very well, because it doesn't have any idea about protein structure. And the pathogenic variants, which are not non-coding, it's actually one of the tasks why we need to develop such models. They are not very well explored, and we don't have very good big data sets to validate. So if anyone has a good data set where we can apply GENA for this task, it would be very interesting to do.
Okay, yeah, thank you. That's very helpful. So yeah, do you think so that maybe these models won't be as good as protein models are right now at doing this kind of?
For coding sequence only, like for a mutation which changes the protein structure and need to predict a sequence, I don't know, but my intuition is that specific protein model should work better with it. But for non-coding variant, this one is an only option, because protein models know nothing about non-coding part.
Yeah, makes sense. Right. Thank you.
Thank you.
I also have several questions. Thank you for your outstanding lectures and for your work. I actually also have questions about mutations, specifically about the parts of genome from other species that you used during training. Did you use specific regions like that are not specific for this species that could be applicable to human or not? You just use the whole sequence of other species?
No, just the whole sequence, randomly fragmented.
Okay, and could you please return to the slide about your first use case?
Promoters.
Yes. I am curious about how this task could help like biologists in wet labs. Why is it useful to predict whether there is promoter or not?
Well, to be honest, we included this because it's a very standard task in benchmarks of models. But I can have several ideas about how it could be useful. So one idea is, when you interpret mutations which are in promoter regions, there are now no tools which are really used in clinics to say that this particular mutation will disrupt the promoter. This is one usage case. And another usage case which also you can find examples in literature that people do something like this, is when you want to design some specific promoter, for example, tissue specific promoter, or promoter with a specific strength. For example, for some biotechnology applications, you would like your transgene to be driven by not too strong and not too big promoter, just an average scale. So for these applications, this also can be used. Instead of picking up one of available promoters, which are just characterized in data, you can invent your own with your own properties based on this model. But I don't know. The good thing about these models is that people can actually try to adapt it to their tasks. So it's just a fantasy of people who work on biological challenges.
Yes, yes. It would be very interesting to see like your model applicable to clinical testing. And maybe there are examples, I don't know, I didn't hear much about them, about previous models used for medical purposes. Like the whole pipeline with predicting something on DNA.
Yeah, well, I don't know if it's a question, but if it's a statement, I totally agree. I would also love to see this in clinical practice. Also, there are many challenges towards translating it to clinics.
Yes, and my last but not least question is about interpretation. You mentioned it like limitation, but did you at least try to interpret like attention maps, whether the interaction between the nucleotide regions correlates with real physical interactions between these regions, or maybe interactions between protein or such regions? Maybe you're planning these experiments in future?
We are doing these experiments now, but we don't have a solid result to present at the moment.
Okay, that's very interesting. Looking forward to your results.
Thank you.
I think we have a couple more questions from Ashish in the chat.
Yeah,
About
I can quickly. So one is, somebody asked about 1,000 genomes. I was wondering if you explored adding data from UK Biobank, and the other was in NLP and text. So far, scaling has held up pretty well. The larger the model, the better the performance. So do you think this was like a first attempt, but like, do you think as a community or as efforts in this direction, building bigger models will show better performance?
Okay, I can probably answer the first one, and Yuri maybe can comment on the second one. So regarding the first one, we did not include UK Biobank data for now. There are many other data sets, like, for example, GNOMAD and Exact, to include human variants, and this is a possible direction. Also, maybe not the first priority, because we don't see that this augmentation actually gives a better performance. What we see now that the gain of performance comes from increasing input size, not from augmentation with other species or with human variants. But maybe we are just not good enough in these augmentations. Maybe we need more augmentation or as a training strategy. I don't know. That's the answer to the first part.
About scaling, I can add that we have models of two sizes, 100 millions and 300 millions of parameters. And on equal, if we consider only equal input lengths, then larger model performs better. So scaling in this case works. But and I think that we can train much larger models with better performance, but just we didn't train such models. Another line of scaling is scaling in input sequence length. We also don't know the actual limits of what input sequence, how much of performance will we gain from longer inputs, and how much we can gain from larger models. So these are two different axes in two different directions where we can go to scale models. But yeah, sure, I think we should try to scale models larger.
Thank you. Excellent work. Excited future.
Awesome. I think that's all. I don't see any other raised questions. I love all the discussion happening after the presentation. And I think all I can do is thank you again for joining us today. I think it's clear that the community greatly enjoyed the presentation and the topic as well. So thank you again.
Thank you very much for the invitation. It was a pleasure.
Awesome. Yeah, thank you again, and
thank you.
have a nice day, everyone. I will stop the recording now and see you all soon, and at the next Journal Clubs or in the Discord server. See you. Bye bye.